And we give a nice welcome to Joel Cherry, President of Research and Development at Amyris in North America. Joel made an effort to have a title of his presentation both in French and English, and we agreed he will make his presentation in French or English? English. So Joel, thanks again, and here's the microphone.
Thank you. Can you hear me? Yeah. Thank you very much, and thank you very much for allowing me to attend. I'm not a big fan of big meetings. I was supposed to be in San Diego at Bio this week giving a talk. I hate that meeting because there's simply too many people to be able to talk to anyone and have an intelligent and relaxed conversation. So having the opportunity to come and learn about what's happening in Toulouse was something I couldn't pass up. I didn't know it was going to be 38 degrees centigrade when I said yes, but it's 42 at home. So I didn't do so bad. I have a talk in two parts. The first part is going to be about the technology platform that we've built at Amyris, and the second part is going to be how we deploy the technology platform in pursuit of products. Maybe a couple of words about Amyris. The picture you see up there is the Amyris production plant in Brazil. There are, you can see 3 of the 6 200,000 liter fermenters. So there's a 1.2 million liter capacity at our plant. It's directly connected to a sugar mill, and the sugar is crushed, the cane is crushed into syrup, concentrated, and sent through a pipe directly into our plant. During the off-season, we use what's called VHP and molasses, which is basically regular table sugar mixed with molasses, so we can run 12 months out of the year. And last year it was sold out, so we ran year-round, and we'll do the same this year. And in fact, are expanding our capacity by building a second plant next door with a similar footprint. So obviously, we're a fermentation-based company. We were started in 2004 by some young guys from Berkeley. And their idea really was to demonstrate that synthetic biology can solve some of the world's biggest problems. The first project we worked on was an anti-malarial drug that's been well reported and most people know about, that was done as a non-profit project, so Amyris didn't make any money on that project, and licensed the eventual yeast strain producing artemisinin, or artemisinic acid, the precursor to the anti-malarial drug, to Sanofi, and Sanofi used that to produce anti-malarial drugs. From there, we shifted into fuels and worked many years, really continue to work on production of farnesene, which is a C15 alkene, branch chain alkene, that when hydrogenated makes a great diesel and jet fuel. And it's approved in the US at 35% blend as a diesel fuel and 10% blend worldwide as a jet fuel. And it's probably about 5 or 6 times too expensive to actually be sold. We sell some of it to some airlines and OEM manufacturers, but it's not a primary part of our business anymore. The original project on the anti-malarial compound allowed us to build a technology platform and expertise in strain engineering that was applicable to lots of different molecules, primarily because there are about 60,000 known terpenes in nature, and both of those molecules are terpenes. But what I'm going to talk about as we talk about the technology platform isn't expertise in a particular pathway to produce a particular type of molecule, but in the underlying technology that's important to make strains, produce compounds at scale at a commercially relevant price. So there's 3 things that are really important for that to work. You have to be able, of course, to engineer the biology. That means putting the instructions, the DNA, into the cell that you want to tell it what to make. You then have to be able to analyze that strain in detail and understand the impact of the DNA on the performance of that strain in terms of production of your target molecule. And you need great data handling to be able to understand what that data means and use it to learn. Now, we talk about something called the Nth Vision in R&D at Amyris. The Nth Vision is that I'm so damn good at engineering microbes that when you tell me you want a molecule, I can go in the lab with a single design and make an organism that produces that molecule at a commercially relevant level, and I can take that directly to scale. It's one shot. That's the nth vision of synthetic biology. This process is really just a process of learn. How do we learn faster? How do we do this faster? How do we cycle through iterations of design to learn what's important to be able to engineer biology? A critical component of this is what we call the ABCs: automation, biology, and computing. And when we hire people, we look for people that have expertise across all three of those, or at least some connection to all three of those. Because if you want to run that design cycle as fast as possible with the best quality, you have to employ automation. You have to know some biology, and you have to be able to manipulate the data with good computer systems. Now, sorry, I was singing with Marie last night, so I'm starting to lose my voice. The most important part of this is really the people you hire, and I was fortunate to hire some really great people about 8 years ago at Amyris, and they were really the ones that designed and built this system I'm talking about. This just lays out in a little more detail some of what we've built in terms of what's necessary for design. Of course, you need enzyme design, you need enzyme improvement and optimization as any part of metabolic engineering. You need to have great pathway modeling tools because that's the basis of your designs. And you need to have genome scale models to understand what are the possible impacts of overexpression of particular genes on your particular pathway. And it's those tools that get iteratively better. Your genome scale model, your ability to predict where the carbon is going to flow once you feed an organism is one of the key parameters. On the build side, excuse me, this is where the automation comes in. We've got a very nice system for constructing DNA, putting pieces of DNA together, using a front end that's called Thumper that allows me, even in France, to sit down and type a string of characters in what we call a genotype specification language that describes the pieces of DNA I want to put together and the location I want to insert those pieces of DNA in the genome. That information is sent into the lab. Most of the DNA pieces from the entire yeast genome we already have in a freezer bank. If we don't, the software actually orders it from a DNA synthesis company that works closely with us. And those DNA pieces are loaded onto the robot, and the system takes over. Automatically, those pieces are consolidated, stitched together, transformation is performed, and we get transformed cells to carry through the test process. And on test, we use multiple layers of testing to try to ensure that what we test correlates with the performance of that strain in a 200,000 liter fermenter. Now that's not easy, and it requires lots of testing to develop the correlation model that allows you to have confidence you can go from microtiter plates to 200,000 liters. This wasn't cheap. Amyris has about $1.2 billion invested in it over the last 12, 14 years. And about 500 of that I got to spend. So, and my team got to spend. That's not all capital, of course, that's mostly people. There's been a huge evolution in terms of the value and transformative nature of automation. In 2008, there were some fairly clunky automation systems available. And really it wasn't until 2010 when you got these nice workstations that were easily controllable by an average scientist without a lot of training. And today what we're seeing is that we're getting integrated work cells that have multiple functionalities that are very flexible and that you can build out to do, our current BioCell, which is an Agilent robot that we use for lots of our assays, can run 26 different assays, and they can switch from one to the next just by switching the plates in the program. That kind of flexibility just changes the way you work and allows you to work on multiple different products simultaneously using the same robotic systems. Again, it's all about speed of the cycle. How fast can we do this? And at Amyris, you know, we've expanded. We used to be just a yeast company. We actually started in E. coli, and the first product, the first artemisinic acid strains were E. coli strains, and switched to yeast for reasons I won't go into, and now are in the process with some DARPA funding of expanding that portfolio into a wide variety of different organisms. Now, this is a little bit busy, but I just wanted to show, maybe we just pick one out and say Hensenula polymorpha. Here's a case where to put DNA into 2 sites with one of those marked, meaning one has an antibiotic marker, used to take last year about 24 weeks. Using this new automation systems and improvements in the transformation efficiency through iteration, we're down to about 9 weeks. And for Saccharomyces, we can put up to 6 integrations in simultaneously in a single transformation, unmarked, and it only takes 3 weeks. Now, to give you some sense of this, we're doing this about 1,000 times a week. We can put in between 2 and 12 parts. All of the DNA that we put in is fully sequenced, so there's sequence verification. And we learned a hard lesson about sequence verification when we went back and looked at strains we'd constructed before we did it and found out that about 25% of the strains we thought we'd built actually had point mutations or indels that were affecting the performance of those strains and, of course, affecting our ability to learn by the strains we built. It's about an 85% delivery rate. That varies from organism to organism. In Cerevisiae, it's actually about 95%. And the transformation system is also an automated system. So as a scientist, I can do experiments really without going into the lab anymore. And this is the future. So, you know, those 3, 4, 5, 6, 7 years you spent in the lab moving liquids from one microtiter plate to the next during your PhD, you know, there's a time coming soon when PhD students are going to spend their time in front of a computer and they're going to spend their time analyzing data, not moving liquids from place to place in the lab. What this does for you as a scientist is allows you to explore those ideas that you would usually just push to the side because you can only make 10 new strains every month or 50 new strains every month. You can make 1,000 strains. If you've got 50 genes for a particular enzymatic conversion step you want to test, you test all 50. If you've got 50 more that you think might have the activity, you test those 50. So it really opens up your mind and allows you to collect a much broader swath of data that informs your learning as you work towards a strain that is commercially viable. Now, none of this works without an integrated computer system, and we have over 1 million lines of code of proprietary software inside Amyris, and from the day I arrived in 2008 and really since 2006, all the data that's been generated at Amyris has been archived. What's really changed, and is accessible via these computer systems. What has changed is our attention to the quality of the data and our ability to put meta tags on that data so that we understand what this particular measurement means in terms, and how well we can interpret that data. And again, that's a hard lesson to learn, but knowing your error in your measurements as soon as possible and tracking that as part of your data set is really important if you want to move towards theoretical yield. And I'm not going to go through each of these computing systems. I just don't have time. I already talked a little bit about the genotype specification language. Here's just an example. In the gray box at the bottom, you can see I want to express 4 genes at one locus. And this is just shorthand that I can type in and send to that software package, Thumper, and will result in these strains being built. Now, what this allows me to do is to vary things in a very simple way. For instance, if I want to measure the impact of a particular gene expression on the production of a desired product, I can, with a single 3-character code, say I want to vary promoter strength from here to here, and the software will pick out the promoters that rank from high to low and create those strains for me, and I'll be able to then get 10 strains back that have various promoter strengths at that particular target gene, and evaluate how does that impact the production of the target molecule. Great stuff. And this, I should say, this is something that is publicly available and open in terms of the genotype specification language. Autodesk has adopted this for their front end for DNA design, and the more this gets out, the happier we are, because it will improve with more people. This is just a screenshot from the Thumper construction. I can tell you on the side sometime why it's called Thumper, but it's just a Disney name. Again, this is the software that most scientists use to design strains and to interrogate, to look at what are the components in their strain designs. As I said, everything is sequence verified, and of course, as strains progress towards production, we routinely sequence the entire genomes of our yeast strains. We sequence about 8 to 12 yeast strains a day, and we do it using tagmentation and barcoding. We found out that we could do sequencing a lot cheaper than the manufacturers of the sequencing machines say, by taking 8 genomes, chopping them up independently, adding barcodes to each of them that are different, mixing everything together, and then using the computer, sequencing it, and then using the computer to pull it all back apart. And by doing that, we were able to reduce the cost of sequencing the yeast genome from about $100, $120 to about $8 a genome. So, for $64, we can sequence 8 genomes now. We use the Amazon Web Services for a lot of the really heavy lifting in terms of computational power, and honestly, we've been extremely happy with that. So, you make the strain, you analyze the strain, you know that you've made what you wanted. The critical thing is, are you measuring the things that matter? Now, in a first glance, we care about how much product is being made. But of course, it's only a small fraction of the cells that we engineer that actually produce more of the product. Most of them are misfires, and we want to know why. So we have to do more interrogative omics approaches. We've got lots of nice equipment around mass spec, C13 flux analysis, fermenters to do fermentations, and some custom robotic platforms that do front-end separation in front of the mass spec, dependent on the molecule characteristics that we're interested in. And this is part of a DARPA-funded project that has a front end that basically understands what the separation parameters for any given molecule are going to be, and sort a microtiter plate full of samples into different separation technologies that are the front end for the mass spec. I'd say this is about 70% complete. It's not fully functional yet, but in the next 6 months or so, it should be good. And in the end, it really does come down to how well do you understand what's happening inside the cell. We've built whole cell metabolic models for all the organisms I showed. Some are really lousy and some are really good. Yeast is great. But they're continuously improving. And again, the whole point of this is to be able to learn what is important and what isn't important. We use these metabolic models. Sorry, I think I had a duplicate there. And map them onto this program called Escher, which some of you are probably aware of, that is just a really nice tool for visualizing data that comes out of your omics analysis. In the upper right hand, you can see visualizing metabolite concentrations, where the circles are the size of the amount of that particular metabolite, which gives you really a snapshot about bottlenecks that is super easy to look at, and you can look at hundreds of these in a row and quickly understand impact of engineering. All right, so all that engineering stuff has to get to a 200,000 liter fermenter, and it has to lead to commercially relevant product. So this says we can make millions of strains per month. Well, the reality is we can make on the order of 7,000 to 8,000 strains rationally designed a month. Most of the others are random mutagenesis. We still use random mutagenesis in sequencing, primarily as a lead generation tool to know where to put our design power in the next round. We're screening about 120,000 strains per month, primarily in 96 well plates, and then have 72 half-liter and 2-liter fermenters to do further analysis. In Emeryville, we have a 300-liter pilot plant. It has 4 300-liter fermenters, and it's mirrored by a pilot plant in Brazil that has exactly the same fermenters and controllers. So when we do a process transfer, it goes to Brazil, and they use fresh cane syrup, and we use old cane syrup. Otherwise, it should run about the same. And then we typically go straight to 200,000-liter fermenters. We don't do any intermediate scales. The 300-liter is run not really for strain performance, but primarily for separation performance, to understand the separation process and parameters. So all this ties together in the pursuit of learning. And that is driven by having really robust automation and computation systems for analyzing data. Thus far, we've got 15 products successfully commercialized. That's from 5 separate fermentation products. And some of the partners that we have are shown here on the right. Now, what this means in terms of our ability to engineer strains, we've written some papers and are fairly well documented on the farnesene production strain that we're using currently in Brazil. But just to give you an idea, that particular strain has about 1% of its genome altered relative to the starting point strain. Now, that's about as different as you are from a chimpanzee, or certainly more different than this Great Dane is from that Chihuahua. So we're really making new strains. We're changing a lot about these strains and how they handle carbon flux. To get to a commodity-based product, this is absolutely necessary. To get to a fragrance compound, probably not. You just don't need to be that price conscious. The other thing this has done for us as we've transitioned more and more products through full-scale production is allowed us to really look at this correlation between 2-liter fermenters, 96 well plates, and 200,000-liter fermenters. And this is the table, or what is that? That's a chart? No, that dot plot on the left, shows you performance of strains at 2-liter, 300-liter, and 200,000 liters for 3 different fermentation strains. You can see that in the last iteration in strain 3, very few 300 liters were run simply because our confidence about transferring to scale got better and better. But statistically, there is no difference between the performance in a 2-liter fermenter and the 200,000-liter fermenter. The other thing all this automation does is allow us to get to products faster, which is what our customers really care about. On the right, you can see in the first one is an example with farnesene. And this, I could have added another bar. We're on our 5th product now that also took about 6 months to go from idea to it's in commercial production. It's being transferred to commercial production. Okay, so that's the technology. That's what drives it. We can talk a couple minutes about applications. First, we'll start with a business model. What we found is that the identification of a target molecule isn't something that we should be trusted with as researchers or as scientists or even as small companies. The partner needs to help you select that target, and to make sure the partner really means they like that target means that they're willing to spend money on it. So usually what you're doing is you're trying to identify the problem the customer has in sourcing, supplying a particular target molecule. And they're willing to pay upfront for that most often. Now, we typically do not get paid on a routine basis after an upfront payment. We get paid on milestones. So we deliver defined milestones to them in a defined timeline. And there's penalties if we're late, and sometimes we get extra if we're early, but not very often. Then we produce the product, usually with a very small margin on the product, but we share on the revenue on the back end. Now what this looks like in reality is we get a lot of collaboration revenue on the order of between 30 and 40 million per year now. In 2016 it was 50 some million. So the majority of our revenue actually came from collaboration payments from our partners. Now what changes is that as those products that we've developed enter the marketplace, they start paying us back because we share in the margin. And there's a delay between when we work on the strain and when we start to see the margin. But you can see that collaboration's revenue basically stays the same, and then these various product areas start to grow and deliver revenue to Amyris. One of these is personal care, and that's where the cosmetics and flavor and fragrances fall. We see a lot of attractiveness in skincare and active ingredients for cosmetics. And we've had some experience with this because farnesene has been dimerized and hydrogenated into a molecule called squalane. And squalane is an existing emollient that was already on the market that we put into the market as a direct-to-consumer brand, so B2C. And Amyris launched a brand called Biossance. And Biossance has now, I think, 6 products, all of which contain various levels of squalane as an emollient. And the flagship is pure squalane oil that's used as stuff for your face. Just recently, Sephora took this on, and sales have really picked up. It's selling quite well. We did this primarily to understand the market better and to understand our customer needs better. We never stopped selling directly to the squalane market that's in the B2B business and continue to do that. And in fact, there's about 400 brands that now contain squalane in their products, some of which are here. Last year, we also announced an exclusive collaboration on cosmetic actives with Givaudan. That's up here on the right. And we formed a joint venture to produce squalane with Nicol Group that runs what used to be a CMO operation on the East Coast in the US and is now an Amyris Nico JV-owned production facility that converts farnesene into squalane and farnesene into a couple other products. So over the past 14 years, we've shifted from being, a company that started with some money from the Gates Foundation to make an anti-malarial drug and moved into fuels and got beaten up by low oil prices and extreme difficulty with making commodity products via fermentation, to a company that now has 10 products that generate at least $1 million in revenue and continue to grow. I think we're incredibly proud of the technology platform we built and feel like it has a lot of other places it could go, which makes talking about Amyris or any synthetic biology company difficult because you can do almost anything. You can make almost anything. To explore that, last year we were awarded a contract with the DARPA to explore what we called the MIGS to KIGS idea, and that is DARPA wanted to make 1,000 new molecules as starting points for new materials, primarily for military use, new clothing polymers or reflectives, tapes, ship coatings, those sorts of things. So they came to us and said, are you interested in this program? And we said, well, just making those compounds isn't enough. You need to make enough of those compounds so that you can actually test them. So we called the program MIGS to KIGS, and we promised DARPA we would make 450 molecules over the course of 4 years, and that 10 of those would be at kilogram scale by the end of 4 years. Last year, and we do that in 3 different organisms. Last year, we made about 10,000 designs targeting 537 molecules, and we're successful at making 164 of those. They're shown, some of those are shown here. This is a dendrogram showing chemical relatedness, and we intentionally picked chemical classes that are widely dispersed, some of which are extremely difficult to make biologically, and have been extremely heartened by the success. I mean, some of these we're making at multi-gram per liter in the first design. The other exciting part about this is it's allowed us to further the whole idea of removing scientists from the laboratory by building a system that allows for closed loop design, test, build, and learn. We call it the automated scientist, and we can now put in a chemical structure, a CAS number or a chemical structure, and get back a first iteration of design. And what we're working on, and in fact the 500 I talked about were designed in that way, but the next step is to take the automated scientist and teach it how to interpret the data so that it can do the next iteration of design, so that I can sit in my office and have a glass of wine with Pierre and not have to do anything. That's the end. So thank you very much, and I appreciate your attention.